Hello, everybody. Welcome to the session of Cultivating Openness, Reproducible Data Acquisition. I'm the moderator today for the session, and my name is Jing Wen. I'm an assistant professor in the Department of Radiological Sciences at UCLA. Before we get started with the exciting talks, just a few reminders. Our session is recorded and is broadcasted live. If you have any questions to the speakers, or if you have any comments you'd like to share, please feel free to post them in the Q&A. And we will go over them after each presentation. You're also welcome to upvote any questions you like. We have 3 speakers today. The speakers are Professor Hongjian He from Zhejiang University, Dr. James Cody from Peter MacCallum Cancer Center, and Dr. David Waddington from University of Sydney. Without further ado, let's welcome our first speaker, Professor He, who will be presenting on measuring brain microstructure using multi-compartment T2 model.
Okay, thanks for the introduction. Thanks for the invitation to this wonderful event. So I think this is my second time to join this event. Last year, I also be the audience to listen to a lot of wonderful talks. So today I'm going to share some of our experience on measuring brain microstructure using the multi-compartment tissue model. So I hope everyone can hear me clearly.
Yeah.
So our as everyone know, our brain is a very complex organ with both in its function and structure. So MRI is one of the most useful technique for our brain research because it can provide a very excellent soft tissue contrast, for both very helpful for morphometrical and structural and functional studies. And most importantly, MRI is a non-invasive and a very economical and practical tool. The little cartoon I show in the below is how MRI works. Due to the time list, I just skip this part, and I hope everyone already know how it works. Then the second thing is the compartmental tissue model. This is the key factor in this talk. So in our brain tissue compartment in MRI, we usually consider it consists of the non-water compartment and the water compartment. The non-water compartment, including the myelin and non-myelin, we say it's non-water because it's a macromolecular tissues. The other thing is the water, including the we all know myelin water, non-myelin water, which is the intra and extra axonal water, and also the CSF regions. So in a compartmental tissue signal, we usually refer to the relaxometry, which is T1 and T2. And sometimes we also mention the probe density sense, which is close to these relaxometry measurements. The other thing is you will use a lot for the diffusivity of this water. It's a very important property of in MRI studies. The most important part that we want to measure is the signal fraction of each tissue contributes to the total signal we measured. So this model has already widely used in both brain science and disease studies. Because the T1 and the T2, the utilization time is very sensitive to our microstructure. So one idea is just we can very quickly to mapping the relaxometry of our brain. So one technique developed by Ma and Ma Dance Group, which is published in Nature Journal in 2013. It's a MRI technique which can simultaneously mapping for both T1, T2, and probe density is very useful and advanced technique. So in my group previously, Dr. Liao Chongyu and Cao Xiaozhi, they are now in Stanford University. When they are still in, they still work in Zhejiang University, developed the faster 3D whole brain MRF technique. With this technique, we can we can simply mapping the 1 millimeter isotropic T1 and T2 information in 5 minutes. And if we want to achieve sub-millimeter resolution, it may increase 1 minute later, 1 minute more. This is a very nice and a very quick, very fast technique. So we can use it in some disease or some brain science studies. For example, this gives us the illustration about our T1 and the T2 mapping in different resolution. The SNR and the image quality is very nice, and the most importantly, the quantitative value are very accurate. So as I mentioned, this MRF technique can simultaneously mapping the T1 and the T2 values in voxel-wise resolution. And the one application we did is used for the epilepsy patients. But I want to mention by that time we still have we are still using the 2D version. The previous version I mentioned in last slide is the 3D version. So this is the case we show in using the MF to detect detecting the epilepsy patients to detect the lesion area. So this is a healthy control patient, healthy control, and this is the one example for unilateral patient. You can see the diseased region, which will have very different T1 and T2 value compared to its contralateral normal region, or you can also compare this value to the normal control subjects. So basically, the idea is once you can measuring voxel-wise T1 and T2 values, you can just diagnose the disease area and detect the disease area by just a threshold of the T1 and the T2 values. It's very nice and very easy to operate. So the idea behind this study is trying to indicate that when there's some pathology change related to the sclerosis in the epilepsy patients, though the macroscopic structure will just change, and they will also change the T1 and the T2 values. But this change is unfortunately, it may or may not be observed in the weighted image because the low contrast to image ratio or low signal to noise ratio. So this is our first try with the MRF technique. So very recently, this is just early view paper work done by Xiaozhi and Chongyu. So they after they moved to Stanford, they developed this DTI and the MF technique for rapid high resolution measuring T1, T2 pearl density, and also the DTI metrics including ADZ value and FA maps. So this is the sequence diagram, but due to the topic of our this session is the reproducibility, so I just skip this part and briefly show the results. So this is the T1 map in 1 millimeter isotropic resolution. This is a T2 map. This is a pearl density map, and very nicely, it's the first time to also simultaneously to acquire this ADZ and the FA value with the B value equals to 600. So if you are interested in this work, you can go to this early view paper in MRI journal. In my group, we actually also have look at the question in the other direction because we are interested in the compartmental signal. So one thing is we trying to mapping the myelin proton signal. So we want to develop a unique sequence with ultra-short TE technique, which can capture the myelin signal itself. So with this signal, with this technique, we can do one whole brain, but like close to 2 millimeter isotropic resolution and it requires like 15 minutes to get all the signal. The idea behind this sequence is for here we are not only interested in the water signal, including marine water, non-marine water, and the CSF signal, but also very importantly, we wanted to map get some idea about get some signal from the myelin itself. The myelin protein is macromolecular with a very short T1 and T2, especially the T2 values is close to millisecond, 1 millisecond. So this signal is not available in conventional imaging MRI imaging technique, but it's available with the ultrashort TE technique, which already shown by Du Jiang's group, and they did some very pilot study in 2020. So in our work, we want to simultaneously measuring both the myelin signal, the proton signal, and also the water signal. And this is achieved by using a dual echo acquisition strategy. So with this acquisition method, we can simultaneously measuring multi-compartment tissue signal. So as we show here, this is a myelin water fraction. This is the conventional T1 and T2 mapping. All in is physical units in milliseconds. And this is addition to these three signals. We most importantly we can also get the very nice myelin proton fraction mapping. So I think this is the first time we can get these two signals simultaneously in one time, in one scan. So we also have some preliminary tests on the multiple sclerosis patients and show quite significant detection power in to get the to locate those diseased regions. So for the previous slides, I just show you our progress in the related to MRI signal, which is indicating the T2 signal is related to is very sensitive to the to those microstructure changes or microstructure information. But earlier than that, we also actually also the other techniques can do this part of the job. For example, the multi-echo technique and together with the multiple exponential fitting. No matter you are using T2 multi-echo spin echo to measuring the T2 decay or the T2 star decay using the multi-echo GRE sequence. You can get some information like as we show here because you can get the signal fraction from the for the myelin water and also for the intra and extracellular water because their T2 values are very different from each other. Then if you can using a nonlinear fitting to get this equation be fitted properly, then we can it's possible to get some idea about the fraction of myelin water and also the other water signals. So this is quite popular in the last decades, and it's also very useful for now to measuring the myelin water. So for here, we probably all the audience should have some idea like the T2 value is quite sensitive, or maybe also the T1 values has some sensitivity to the microstructure information. So the next part is we are going to switch to another popular imaging technique. Is the diffusion imaging. So in the diffusion imaging, a very conventional sequence is we use spin echo and the pulse gradient to measuring the water diffusive motion information. So this is the diffusion gradients we applied during between the 19 and the 80 IF pulse. So with these diffusion gradients, we can get different the signal in different B values. So if we fit the B value and we get the signal, you can see here we get some signal decay, which is follow very nicely exponential decay. So when the signal drops faster, that means the motion is very fast. So in this diffusion imaging, it's a very important thing is to measuring to estimate the diffusivity value. A popular way is as I show here is using the diffusion intensity imaging, which also called as known as the DTI. In DTI, we measure different direction of the diffusion gradients, and we can get some estimation, measuring the signal in each direction, and get a very nice estimation of this D value along the direction. And we can even get the calculate the tensor matrix and get the water the reproducible direction which water molecule moves, and we can even get the idea how the fiber water axon fibers are arranged using this technique to check the water molecular directions. So, with these FA values and the diffusivity values, it's just like scalar measures to indicate the tissue microstructure properties, which is close to our topic today. The FA and the MD values is very popular as I wish, but their computation is quite easy, quite simple, as we show here. If we are interested in the average diffusivity value, which is an MD value, we can just use in the lambda 1 to 3 in 3 directions and do the calculate their average value. But for the anisotropic values, you need to use this equation to see how different from in each principal axis direction the lambda value divert from the other two. So in diffusion MI, if we come back to this plot, we are actually not sensitive to the non-water signal, and we are not able to capture. Usually, we are not able to capture the mining water signal because it's too small. And we actually look at the non-mining water, including the intra-axon and the actual axon water signal. And also we get some free water signal contribution to the measured signal. So this is give us a very simple diagram to show this is the axon. It's magnetized very well, as shown here. The black guys. So if we just show a simple diagram on the right, is in this yellow guy is the intra-axonal diffusion signal. This gray guy is the extra-axonal diffusion signal, and the blue guy is the free water, as I mentioned here. So basically, this is trying to get the total signal from these 3 compartments. So we're using this multi-compartment tissue model to understand its signal evolution. So if we come, so the first thing is we need to, because each compartment or each part of the tissue, the diffusivity value in terms of the motion ability of these water molecules. So the diffusivity value for each compartment is very different from each other. Then one thing is we can, if we in our imaging resolution, we actually for a particular voxel, the signal is the total signal for all these compartments. So it's harder to tell which part is contributed more than the other one. But But when you have multiple b value instead of just one b value, you can calculate the FA value for each sorry, for each b value. For example, we can calculate the FA value using the equation I just showed here. Basically, if you only interested in the FA value, you don't need to. You only need the B0 and the additional B value. For example, B1000. So the 2B value will give you some estimation about the FA value. So the idea is when you're using 1000 B value to calculate the FA value, then you're using the 3000 B value to calculate another FA value. Then you do the pair t-test to compare these two FA values, even though they are come from the same brain. The result is showing here like the small b value for 1000, it will be much larger than the FA value you calculate from the 3000. So this result is you can just repeat it very easily using other pairs of b value, for example, 1000 and 2000. And you can find the related result in this paper in Hubert mapping published by my student Yao Junye. So one thing is, so this is the FA dependence on the B value we can very easily replicate in the diffusion MRI. Then we are interested in what's the contribution for this difference measured in the for this microstructure metrics. So one thing, one factor mentioned already by Derek Jones' paper in MIM 2004 is already they discussed a lot about the noise effect, which is a noise floor. So the idea is when you apply the higher diffusion gradients, the signal will drop very quickly, but the noise is still there. When we get the image, we actually have to take the absolute value to get the magnitude image. Then this is no matter the noise is negative or positive in the but the final result will be just give you some absolute value. So this is a noise floor defined. But in our study, most importantly thing is we find this difference in FA value is also correlated to the histology showing here is the NDI, that means the neural density information. So we conclude that this difference of intra and extra neurates, which is a compartment divisivity, can also contribute significantly to the effect we observed. So this is the B value factor is a factor we know is very important in diffusion MRI. The other factor, the echo time, is a second factor I want to emphasize. So the echo time I want to just to mention a little bit. The echo time value is actually in a typical diffusion MRI scan, it varies a lot from the around 70 milliseconds to even 120 milliseconds. Depends on the performance of your gradient system. Then the T value usually gets higher when we want to achieve a higher B value and a higher voxel resolution, spatial resolution. So the T value we show here is a time between the 19-degree pulse and the echo time. So the recent high performance gradient allows us to reduce this T value. For example, the Prisma scanner we are using in our center, we can get the minimum T value to 68 milliseconds. Even the maximum B value and the maximum B value can reach the 10,000. So it's very nice to have important for nowadays to get a high performance gradient system to do the diffusion imaging. So for now, that's also the reason we want to get the multi-shell high angular resolution high diffusion imaging. So we did some preliminary study. This is still published data. We acquired in different scanner using the it's best optimized protocol. But the thing is, the T value for different scanner will just vary a lot across different scanners because the performance of the gradient system. So we do some variance test, and it's important thing is the interscanners variation is always be larger than the inter-substrate variance in different vendors. Though we can also do some sensitivity analysis to see in one scanner like our Prisma scanner, we can analyze the three factors, including the T value, B value, and the B0 number. And we found that the T value and the B value are two main factors of the variance. So we already emphasize the importance of B value. Then we should pay more attention to the T value for later. So the T value is one of the, as we all know, one of the most fundamental and important imaging parameter we can see in any sequence. So with longer T, the MX, the transversal magnetization will just decay more due to the transversal relaxation. So the decay ratio varies significantly among different tissue types according to their T2 values. So this is one tissue, this is the other tissue. That's why we get the T2 contrast in our imaging. So the choice of our T usually determines the T2 contrast of MR image. But then we come to our diffusion imaging. So in the diffusion imaging, each tissue will, for example, we show on the right side, we show two different type of tissues with different T1 and T2 value. So you can, in diffusion imaging, we select a very long TR value, for example, 3 times to the T1 value of the tissue, then you can get all the T1 signal recovered. Then in that case, the T value will be a crucial factor, but it's often overlooked, as I mentioned in diffusion MRI. So we consider the tissue heterogeneity, as we mentioned. There, if in one imaging workflow, we have instead of have a single type of tissue, we have multiple tissue types. Then with the compartment T2 and the diffusivity model, as we mentioned. So now the simple equation here showing the signal exponential decay will not hold, and we need to correct it as the equation below. We consider different tissue type have different T2 value and the different diffusivity value. Then we get the total signal for both of them. Then if we do the DTI study again, we are measuring in different echo time, and we do the calculate the FA value. It's very easy to get this linear shape, both in the monkey brain and in the human brain. So in 2018, we found this, and we trying to do some simulation to Monte Carlo simulation, trying to explain the results. And we found this linear shape is just due to this compartment T2 contribution. So we can assume the short T2 of our compartment is around 60, and the long T2 tissue is around 100. So in that case, you can just fit the signal, get the simulation result very similar to the result you get from the real experiment. So the conclusion is the microstructure bias of the T dependence. I say the T dependence of the FA value in the FA observation is due to the microstructure contribution. So the similar idea we also tested on the advanced signal model, which is a NODI developed by Dr. Zhang Hui in UCL. So this the NODI is a 3-compartment model of diffusion signal. It's consider the intranuclear, extranuclear, and the CSF region. So we do a similar study. We measuring different echo time, and we fit it with each echo time. We fit the NODI model using the the toolbox, publisher toolbox. But basically you can see the interest fraction is increasing very significantly when you increase the TE value. Also, you can see some change in the isotropic value. But this change is not obvious, is subtle in the ODI values. That means ODI is not sensitive to this archetype. So the again, the compartment tissue specific T2, we our hypothesis is the compartment T2 will resulting in this T2 weighted contribution and causing the T dependence in this parametric mapping. And we also do the simulation to understand the this phenomena, and we can replicate using our simulated data to show the experimental findings very easily. And more importantly is if we want to correct this T dependence, we basically we find this needs this correction needs 2 or 3 ergo time. And this is a result, simulation results to show how 2 or 3 ergo time can correct the this dependence. And similar results we can show, we can find in for the in vivo human data. I think I just ran out of time, so I just come to the final results, which is a summary. So in this talk, I just briefly show you the cutting edge quantitative MRI techniques. For example, the MR fingerprinting sequence. With this technique has higher speeds and higher resolution in imaging acquisition, and it has the ability to measure multiple tissue information simultaneously, especially the DTI MRF and the UTE MRF. So, we hope these techniques can improve the reproducibility, and with because they can get us some quantitative measurements of the brain structure. The second thing is the relaxation time, which is a T2 sensitive, is very sensitive to the microstructure information with either direct or indirect detection methods. And the voxel level T2 mapping can be achieved with faster 3D MRF technique. And also the compartment T2 at the subvoxel level can be achieved using techniques like UT MRF or the MR, the mining water imaging using multi-echo GRE or spin echo techniques. So the final insights on the diffusion MRI is the compartment T2 contribution will contribute to the T dependence on diffusion metric, no matter it's the DTI metrics or the NODI metrics. And some biophysical factors, this is some biophysical factor to be considered because it combines both microstructure information and the physical TE factor. So this is something I think we should consider in multi-center diffusion MRI studies to get the better reproducibility. So with that, I want to thanks to my team and also the graduate students from my group, including Xiaozhi, Congyu, and Li Qing, Zihan, also Li Mu, Gongting, Qiqi. They did a lot of wonderful work in diffusion MRI, and in mining water imaging, which is Dr. Wu Ze. So we also have a lot of collaborators, including in Rochester University, Jian Hui, Professor Jian Hui, and Zhang Hui in UCL group, and also the FA dependence work in we collaborate with Calamira in Oxford and Ben in Oxford. We also get some funding supports, including the as I show here. So that's all my talk. Thanks everyone.